All right. Well, thanks everyone for coming along online. A little bit of a change of pace for my normal topic of conversation at these meetings. Typically, you'll find me talking about some of the work that I do with native mushrooms, but I thought today instead what I might actually talk about is what I spend the vast majority of my time actually doing as a medicinal chemist, which is working on novel solutions for treating invasive fungal infections, which is what my PhD was in. So a lot of what I'm going to be talking about tonight actually comes from the work I did during my PhD. Okay, so invasive fungal infections, probably a lot of you here would be at least somewhat familiar with fungal infections. Generally speaking, most of you will have had some kind of fungal infection at some point in your life. Invasive fungal infections differ in that they are infections that enter into your bloodstream and infect your internal organs. And they're a huge problem around the world. They're estimated now to contribute to about 3.8 million deaths around the world every year, of which about 2.2 million are directly attributed to the fungal species. And there aren't very many species of fungi that actually cause these horrible infections. In fact, as I've got up here, there's only about 20 to 25 species that are responsible for the vast majority of infections. But they're increasing both in prevalence and also in our drug resistance. And so our ability to actually combat these things is dwindling. So they've increased quite a lot over the last couple of decades for a lot of different reasons. One of those is the fact that we've gotten really good at keeping people alive in the last few decades with medical advancements. And so the populations who are most at risk of catching these infections are people who are immunocompromised. Typically, these are things that are caught in hospital environments. But also with things like climate change and things like the COVID-19 pandemic, we've seen in the last few years quite a significant increase in prevalence of these infections. Climate change is actually quite an interesting one, and I put this slide in here. Some of these slides actually come from a lecture that I give for third year fungal biology. But the climate change thing I think is quite interesting because essentially the reason why climate change can contribute to these is a result of the fact that fungi generally aren't adapted to live in or thrive in environments at temperatures similar to our body temperature. But of course, as the temperature of the world increases, fungi start to adapt to warmer and warmer temperatures, which means that they start to approach temperatures closer to our bodies. And so as a result of that, and then also things like natural disasters, we get an increase of invasive fungal infections, and we also have an inability to manage them as well under those sorts of settings. A couple of years ago, so invasive fungal infections are a pretty underappreciated issue in global health. And it was only a few years ago that the World Health Organization issued its very first fungal priority pathogens list. I think there's been one for bacteria that's existed for a while now. And so they've listed of the species that cause invasive fungal infections. They've essentially prioritized those that we need to be paying probably the most attention to. In this critical group here, there are a couple of species that one of which you might be familiar with, perhaps Candida albicans, which causes things like a lot of superficial infection, things like thrush, but is also one of the leading causes of invasive fungal infection as well. And in fact, I think roughly about 36 to 40% of invasive fungal infections arise from Candida albicans. And then we also have things like Cryptococcus neoformans, which can cause central nervous system invasive infections. Aspergillus, which is a mold fungus and a very, very difficult one to treat. And then things like Candida auris, which is actually quite a new entrant into this list. Candida auris, in fact, only came about in around about 2009. Or it was first reported in 2009. And since then, I think there's been some retrospective work done by the Centers for Disease Control in the USA. And the first known case of that, looking at old patient isolates, has now been identified as one particular isolate in 1996. But between 96 and 2009, there was nothing. And since 2009, Candida auris has popped up independently in, I think, three different continents within a couple of years, completely different genetically. So these are independent emergent infections from this species. And the reason why this one is particularly scary, and one that I've focused quite a bit on in the last couple of years, is because of the fact that it is almost universally resistant to one of the main drugs that we use to treat fungal infections, fluconazole. And we also see quite a lot of resistance in this species to some of the other classes of drugs that we use to treat invasive infections, such as the echinocandins, and even some of them are resistant to all of the classes that we use to treat these things. They're also really hard to get off anything. So in hospital settings, having to clean when you're sterilizing hospital surfaces, quite a lot of the conventional methods used for sterilization don't work with Candida auris, and that's where a lot of these infections are actually acquired. I wanted to give a couple of other examples of some maybe slightly weirder examples of invasive fungal infections. So ones that I found interesting that I've learned about actually quite recently. So these images come from Eva Jasinska, who works at the Department of Mycology in Pathwest QE2 in WA. And this particular species, and I said I was going to try and practice how to pronounce this, and then I proceeded to not do that. This particular species is one that is found in soil. And the reason why this one is quite interesting is because of the fact that, unlike almost every other agent of invasive fungal infection, this one infects predominantly people who are healthy and immunocompetent, and mostly males. I think I read the majority of people who are infected by this are aged in their 20s or 30s or 40s, I think. And the other reason why this one is particularly unusual is because of the fact that it causes CNS infections, which is very rare for any sort of invasive infection. And if you get it, almost certainly most people who get this will unfortunately end up dying. There's not a lot of ways that you could treat this particular type of infection other than surgical resection in combination with different antifungal agents, but most people within a few months unfortunately won't survive this one. And the other one, which also comes, this image also comes from Eva. This other one that I found quite interesting is one of the very, very few examples of a macrofungal species that can cause invasive infection. So most people here will be familiar with this one because you will have seen it probably in your gardens, as Schizophilum commune, a split gill mushroom. And you can, this one can cause invasive infection of the lungs. So most of the time, this one causes invasive sinusitis, but can also cause pneumonia. It's very treatable, but unfortunately, the diagnosis of this is one that is particularly challenging. And then this last example is one that I came across a few years ago, which I found interesting, to say the least. Not a very common thing that you would think of as being responsible for any sort of fungal infection. So this is a case study from someone who decided to inject a tea made out of psilocybe mushrooms that they filtered, as it says here, through a cotton swab before injecting it directly into their bloodstream. And then this person proceeded to get very unwell. And after doing some culturing of their blood, they found that this person had both bacterial infection as well as a fungal infection, which turned out to be because of the psilocybe that they had injected into their blood. Okay. So one of the major issues with invasive fungal infections is the fact that we do not have very many drugs to treat them. We only have a few different classes, so predominantly these fall into one of these three classes. Echinocandins is azoles or polyenes, and I've also got these pyrimidine analogs down here as well. Almost all of them will target aspects of the cell wall or the cell membrane of the fungal cell, and resistance to these is becoming increasingly common. And unfortunately, there hasn't been a lot of research or motivation to really find new targets or new drugs to help get around that issue in the same way that there has been for bacteria, although even for bacteria that there has not been as much research as there probably needs to be. So the first one of these is one of the oldest ones. So these are the polyenes, so these ones here. And so the way that these ones work is they will bind directly to this little molecule that I've represented here, this ergosterol, which is the fungal equivalent of cholesterol that sits inside the fungal membrane. Resistance to these is not very common. And which means that they're a very, very handy drug to be able to use. But unfortunately, they also have quite a lot of associated toxicity. The other problem, so that is to do with the fact, of course, that ergosterol, despite being different to cholesterol, is not that different, and so there are problems where these polyenes can also bind to the human cholesterol as well as ergosterol. And as a result, there is a lot of toxicity with those ones. The next one, which is probably one of the more common ones, these are the azoles, and these ones are ones that can be found worldwide. So they're generally quite effective, although some species, things like that Candida auris I mentioned before, can be quite resistant to them. And so these work by basically blocking. So these also interact with ergosterol, but instead of actually interacting directly with ergosterol, what these do is these will block the synthesis of ergosterol. And so this enzyme here is, we don't have a human form of this because we don't use ergosterol, but and so the azoles work by blocking this. And when you don't have ergosterol in your membrane, essentially what happens with the fungi is that their membranes are no longer rigid, they're very fluid, and they pop open essentially. You can also get those orally available as well as through IV, which is a huge benefit. Most people tend not to like having their medicines injected to them if they can just take a pill. One of the more recent ones are these echinocandins, and so these ones are generally quite good. They're very effective in treating a lot of different fungi, so they've got good broad spectrum activity. One of the major problems with these, though, is that they are not affordable, and because they're quite new, there's not a lot of generic versions available of these ones yet. And so countries and areas such as sub-Saharan Africa and places where there's not a lot of money to be spent on these things, these drugs are essentially just completely unavailable. And then the last one is these pyrimidine analogs down the bottom here. And so these work by basically intercalating with DNA and RNA and blocking protein synthesis. And these aren't really given by themselves because of the fact that they're extremely toxic. These are normally given in conjunction with some other antifungal drug. And so they're not really useful by themselves. And some I don't often don't really include them often on this slide, but these are used in combination therapy quite a bit. So what's the issue then with making new drugs, and why do we only have drugs that target such a limited number of things? And the main thing really comes back to the fact that fungi, much like humans, are eukaryotic, and so we share a lot of the same molecular machinery due to the fact that our genetics are actually quite similar. As a result of that, it means that we have issues of specificity. So meaning that a lot of the targets that might be druggable in fungi, we have human equivalents of, and so they will hit essentially the same thing in people as they might do in fungi. And in a related fashion, is toxicity as well, for the same reason, basically, the fact that fungi and humans have a lot of the same things. And one of the other reasons that they're quite difficult to treat in the same way that bacteria is that when they adhere to surfaces and they live inside the body, they form these biofilms, which are these really complex matrices of sugars and proteins and cells, and they're very, very, very difficult to penetrate with any sort of drug. And so because of all of these reasons, there's not, as much as there might be some research going on, it is hampered in a lot of ways by the fact that it's just really hard to find things that you can actually hit in fungi that won't also hit something in people. And then one of the other big problems is really just the lack of investment. In the same way as it is for mushrooms, there is just a general disinterest by the people who've had money in funding research that has anything to do with drug development for invasive fungal infection or any sort of fungal infection, really. Making drugs costs a lot of money. So this is just to give you an idea of what that kind of process looks like and where I fit in and where some of the work I'm about to talk about fits into this process. So drug design, from start to finish, is a very lengthy process. On the very, very short end of that scale, like you're looking still at maybe 10 years, up to about 20 years to get from the lab into the clinic. And that all up will cost about $1.3 billion. Most of the things that we will make as a medicinal chemist won't go anywhere. There's an extremely high failure rate. So just because, and anyone can find something that is really potent, so something that can kill fungi, but where things tend to fall down is once you start getting into these clinical trials and you start putting these wonderfully potent, beautiful molecules that you've made into people or into animals. And it turns out that things just don't work the same once you start putting barriers in front of them, like your intestines or your blood or the blood-brain barrier, or things like the liver and all of the enzymes that they have for chewing things up. So essentially, by the time you get to this point, and already at this point you've spent probably millions of dollars just trying to get anywhere, roughly about 90% of the drugs that get to this point will not make it to the end. One of the things that we really, really need for invasive fungal infection, though, are new molecular targets. And so over the last 20 or so years with the fungal genomics era, we've had a lot of new targets that have been identified. And so these are, when we're looking at these, what we're really looking for are things that are not the same in people. So fungi do have some processes, some metabolic processes that are unique to them, or at least not found in people. And so we can use those to try and identify new proteins that we can maybe hit with drugs to try and treat infections. And one of the ways in which they differ is in the way that they make branched chain amino acids. So branched chain amino acids are these ones down here, so leucine, valine, and isoleucine. And in humans and in mammals, we get these through our diet, so through food. However, plants and fungi and bacteria and many other organisms do not do this. So they instead will make their branched chain amino acids de novo. And this is what it looks like. So this occurs in the mitochondria of the cells. And we go through this process starting with a molecule of pyruvate, and then with either another molecule of pyruvate or a different molecule, ketobutyrate. This is the first enzyme in this process here is acetohydroxyacid synthase, and it goes through this process to either make your isoleucine, your valine, or your leucine, which are then used for making proteins and whatever else have you. And so today I'm going to talk a little bit about this first enzyme in the process, this AHAS, acetohydroxyacid synthase, which is what the lab that I did my PhD in and spent many years researching. And this is what it looks like. So this is the crystal structure, or actually it's not a crystal structure. This is a electron microscopy image of a fungal version of acetohydroxyacid synthase. And so in green here, these are like these big multimeric units. So there's lots of individual bits that are sort of stuck together. And at the peripheries of this little Maltese cross shape are the catalytic subunits. So this is where the reaction that it performs is done. And then in the middle here, in purple, is the regulatory subunits. This bit in the middle actually controls whether or not these catalytic subunits can do their job, essentially. And what is really useful about this is these structures is that the spot where these reactions take place, which occurs, you can't quite see it in here, but you'll see it in the next slide, where it does this reaction in between the two units of the catalytic subunits is very, very highly conserved. So the amino acids that live here are almost identical between plants and between fungi and bacteria. So this is the catalytic subunit of Candida albicans, AHAS. And so, as I mentioned, when these things come together, the reaction takes place sort of at this interface. So when they come together, they make this little pocket, and inside that pocket lives these what are called cofactors. So we have this little molecule here, which is called FAD. And then this little THDP here, which is bound to a magnesium ion, and you get 4 of these, where these all connect up together and then attach to the regulatory subunit. And that's what the reaction looks like, basically. So your THDP, which is this little guy down here. So this one actually participates directly in the reaction that ends up with these molecules that then go on to make your branched chain amino acids. It turns out actually that we know a lot of this because targeting this enzyme is not a new concept. So compounds that inhibit acetohydroxyacid synthase have been used for the last about 30 or 40 years in the production of herbicides. And so these have found a huge amount of success in agriculture in this area. And there's a lot of different structures that have been developed over the last 3 or 4 decades. And mostly what I'm going to talk about today are the top 2 here. Mostly actually just this top one, the sulfonylureas. And I've given some examples here. So chloromuronethyl is one that we'll see a couple of times as we go through this. But most of these will have like some sort of a bridge in the middle here, and then at either end of them, you'll have these little aromatic rings, and these will sit into that binding pocket that I showed you before, and basically block the enzyme from being able to do its reaction. And in plants, this is a very effective way of controlling weeds. Basically, just kills them. They can't do anything if they can't make these branched chain amino acids. And as I mentioned before, the site where these reactions take place between plants and fungi is very, very similar. And so what our group thought early on was maybe we could use that as a platform for designing new antifungals. If they're the same, then perhaps these herbicides could also work in the same way in fungi as well. So how do we actually test that? Well, the first way that we might look at testing that is by taking those AHAS enzymes from fungi and seeing, well, how well can it actually stop it from doing the reaction? And so we can and have isolated and expressed and isolated these AHAS from different species of fungi. So these are, I think we've done more than this now, but these 4 are the ones that we've worked with probably the most: so Candida albicans, auris, Aspergillus fumigatus, and Cryptococcus neoformans. And then you kick off the reaction. That give it its cofactors, you kick off the reaction, and then as the reaction starts, you then throw some inhibitor at it, and you see how well it can stop. That inhibitor will stop the reaction from continuing. And we can track that using UV-Vis and look at how that compares to the same reaction, but with no inhibitor. And at the end of that, we get all sorts of numbers, but the ones that I've shown here, this is called your Ki, so the inhibition constant. And essentially, a lower number means it is binding more strongly to the enzyme. And so for comparison, Arabidopsis, which is a model plant organism, you can sort of see here, so these two representative examples from the sulfonylurea, and this one's from the trizolopyrimidine class of herbicides. They have reasonable, like pretty good inhibition in the nanomolar range. Nanomolar is quite good for these. But in fact, these same herbicides can actually inhibit the fungal enzymes even better. And what we found in working with these, and in particular working with these trizolopyrimidines here, is that when they bind to the enzyme, they don't just bind it and leave, which is what happens most of the time when something binds into an enzyme. It's not a one-off process. It doesn't just bind and stay there and then that's it. Normally it's in equilibrium, so they'll bind and then they'll go away. But actually, some of these herbicides, when they do that, they will actually modify the cofactors that are inside the enzyme by trapping a molecule of oxygen in there. And over time, the inhibition of that enzyme increases to the point where that enzyme is functionally inactive. So even once the inhibitor, the herbicide, leaves, the enzyme still can't do its job because the cofactor that's sitting inside there is no longer functional, and it takes quite a long time for those enzymes to then spit that out and get a new one and be able to do the reactions that it would normally do again. The other way that we will test this is by looking at how effectively they can stop the cells from growing. So we incubate cultures in these 96-well plates of our different fungal species, so Cryptococcus, the Aspergillus, Candida, and Candida albicans, as well as Candida auris. And we incubate those with different concentrations of various herbicides. And I've just given just some representative examples again of some of how these herbicides perform in the fungi. It's reasonable inhibition when you compare these, except for the case of Aspergillus fumigatus, which is kind of a notorious one to kill. As I can tell you from experience, I have a very hard time killing Aspergillus. I'm very good at killing these ones, but Aspergillus is something else. When you compare these, though, to our frontline drugs, so things like amphotericin or itraconazole, which are commonly used in treating these infections, for the most part, they're sort of within range. So our Candida auris and albicans values are pretty good when you compare those to our standard drugs against the same strains. Aspergillus, as I said, less so, and Cryptococcus was reasonable, 19 micromolar as opposed to 1.7-ish micromolar is within a couple of dilution steps. So it's not great, but it's a good start. So this is really good. So what I tell you then, if you were to ever get an invasive infection, to go and drink herbicide, probably not. These herbicides actually are not toxic at all. And in fact, chloromiron ethyl, the one that we have done a lot more work with, so we've done some mouse work with chloromiron ethyl as well, and it is extremely non-toxic. And in fact, I think it is less toxic than table salt. So it's not going to kill you. Except if you're using it to treat an invasive infection, in which case the fungus might still kill you. So we did some mouse work a few years ago, so where we infect some mice with Candida albicans, and we treated that with either nothing or fluconazole or one of the herbicides, chloromiron ethyl, and while we were able to detect decreased fungal load in most organs except for the brain, which is important. But by the end of the experiment, the number of mice that survived compared to fluconazole was considerably less. So only about 30% of the mice survived at the end of the 40 days, compared to fluconazole, where the survival was 100%. And so what does this tell us? Even though we know that so CE is very clearly a potent inhibitor in vitro, especially of Candida albicans, and it's very good at binding to AHES. And we also know that AHES is necessary for Candida and other fungal species as well, like Cryptococcus, to be able to infect. And also to be able to survive. If they don't have AHAS, they can't live. So then why then do these herbicides not work when you start putting them into mice? And basically what this tells us is that even though they might be very good at doing that, they're just not very good drugs. And so I mentioned before that one of the major hurdles in developing any sort of drug is the fact that mammalian systems are just very, very good at metabolizing things and excreting them, or just stopping them from getting to where you want them to go. And in this case, that is what we see. So the AHAS herbicides are just not very good when you start putting them into animal models, meaning that they're not very druggable. But we do know that AHAS is an essential enzyme for their survival. And so perhaps if we then make some changes to that structure and we can improve that, its ability to get to where it needs to go, or to not be metabolized, then perhaps we can come up with something that is both a very good inhibitor in vitro as well as in vivo. And thankfully, we have a lot of things to go off when we're thinking about this. And so AHAS, we have a lot of structural information for thanks to X-ray crystallography and things like electron microscopy, so cryo-EM. And so we can then have a look at how these herbicides are binding into those pockets, and have a look at where maybe we might like to make some changes to design new molecules that will fit into the same place, or maybe even fit better. And so this is chloromuron ethyl sitting inside a crystal structure of Candida albicans AHAS. And so it interacts with, so this tryptophan residue here interacts with this ring down here. And there are a few interactions that exist sort of between where this nitrogen is and these oxygens here to other residues like this lysine, and there's an arginine down here that interacts this way. And so we have to think about these when we're trying to design new molecules. But of course, one of the first questions that we want to ask ourselves is what the problem actually is with this molecule. Like why when we put it into a mouse, does it not work in the way that we want it to work? And so again, chloromuron ethyl is the one that we did the mouse work on. So this is the one that, so this is where actually I came into my PhD, was at this point of the project. And so what we wanted to know first was where perhaps the problem is in the structure, and where we might like to make some changes. And so this, the first thing that we identified in this structure as being a potential liability is this functional group in the middle here. So this is called a sulfonylurea. And the reason why this is potentially a problem, it does appear in other drug molecules. In fact, it's a very popular motif that you'll see in diabetes medications. But in physiological conditions, this nitrogen here becomes charged. So this hydrogen is removed. And this can essentially fall apart. And in fact, when we have these herbicides in the solution, that the solvent that we normally put them in when we are doing this work, it is good for maybe a couple of days before it just breaks down completely. And so there's a potential metabolic issue with this part in the middle here. And the other issue is that these molecules are quite flat, and so they are also very just insoluble. So generally speaking, these sulfonylureas can actually help with things like water solubility, which is an important thing when you're trying to design new molecules for drugs. But in this case, I think because of the fact that it's flat, and also because you have these aromatic rings attached on either end of it, it's just brick dust, basically. So when I started my PhD, the first thing that I did was I took this structure and I came up with a few different modifications to the atoms in this bridge to try and get rid of that issue. So try to basically just get rid of the sulfonylurea, make it into something else. I had a look at about 30 different analogs of those computationally. So where I decided on particular changes, I then modeled those structures to see what they would actually look like in 3 dimensions. And then using our crystal structures, I then fit those back into the AHAS to see if it would bind well, and would it still have the same connections that we knew that it needed to have to be active. So there were 30 that we had a look at. Ultimately, we chose 1 or 2 of those where we basically got rid of this nitrogen atom in the middle. So this then just became a carbon in the middle. And then from that, since I've started until now, I think we're up to about 150 or 200 now compounds that have this new bridge combination in the middle and have differing groups around either end here to try and maximize the potency and solubility and a few other things as well. And so we're actually pretty much at the end of the where we've gotten to with this project, and I just wanted to give one example of one of the things that we made, or that I made during my PhD, to show you how it compares and how we've been able to try, at least in the first part, just to make sure that we're making things that are potent. And this one here has been the standout of all of the molecules that we've made so far. And actually, this is one that I made very, very early on in my PhD. And I think since then, there's been a lot of work in the group where I did the synthesis in making quite a lot more analogs. But this one has always just been the best. So this one has a very, very, very tight inhibition of the Candida albicans AHAS. It also binds very well to some of the other AHASs as well. And the minimum inhibitory concentrations of these, so this I should have mentioned before, these are the lowest concentrations required to completely inhibit the growth of these cultures. And for this molecule here, for 58, we found that the MICs of those are actually, in some cases, even better than some of the herbicides that we looked at, and as good or better than some of the standard drugs that we use as controls when we're doing these studies as well, except, again, in the case of Aspergillus fumigatus. And I don't have data for Cryptococcus here either, so I don't think, I didn't really test it too much in my PhD, and I don't think they've done too much since then. But the other really good thing about this is that it is not, the inhibition is very good, but these don't just stop the cells from growing, these actually kill the cells. And they show this time-dependent inhibition that I mentioned before, where they bind into the enzyme and they stop it from doing its function, and then when it leaves, the enzyme still can't do anything because it's had this modification basically take place inside. And it does this to a considerably greater extent than any of the herbicides that we've looked at. This one, I think, it's like 10 or 100-fold, I think, better in terms of its ability to just kill the enzyme compared to some of these other ones. And we're in the midst at the moment of looking at doing some in vivo studies on some of the synthesized compounds that we have. These do have improved physical properties over some of the herbicides in terms of the sorts of parameters that we would look at when we're designing drugs. So things like, will it be able to pass through cell membranes? So we look at things like lipophilicity and solubility, and any other metabolic liabilities that might be there. We can essentially take these molecules and throw them at enzymes that we might find in the liver to see how they break up. And it turns out that these ones are actually pretty good, and they don't seem to, this one in particular doesn't seem to have any issues in that regard. And they're also, as with the herbicides, not toxic as well. So there's a lot of work still to be done. This is all very, very beginning stages, but we've been able to show that the herbicides themselves are a pretty good platform for potential new drugs for invasive infections, but also that these new synthesized molecules, using that as a basis, show quite a lot of promise as potential new antifungal agents that don't seem to have, at least from what we've seen so far, any significant toxicity or any other issues that might prevent it from going into preclinical studies. Okay. And so that is pretty much all I had to talk about this evening. So as I said, this was all stuff that I did in my PhD. So there's me during at the very end of my PhD in 2019. And so this is Craig Williams, who is my PhD supervisor. And I also worked in the labs of James Fraser, who's a fungal geneticist here at UQ, and Luke Goodart, who has really spearheaded most of the work around this AHAS. He's the one who's done all of the crystallography and electron microscopy and everything else. And also like to give my thanks to the funding agencies for the money, and also to our technical staff who helped me a lot during the synthesis phase of a lot of the stuff that I did. And thank you all for your attention.
Thank you, Kylie. Awesome. That's amazing work that you're doing.
Thank you.
Thank you. I see there's says Alberto says fascinating as always, Kylie. Thank you. All right. If there are no questions, I'll stop recording. Oh, here we go. We have a question. Ellis has said, just regarding Aspergillus, I read just recently that a likely contender for the curse of the Pharaoh's deaths was in fact researchers inhaling the spores from that.
Yes, I think I might have read the same thing. Yeah. Yeah, it's very interesting. It's a very, yeah, Aspergillus is an annoying one to work with and a difficult one to kill.
Yes, sounds like it.
Yeah.
Great. Are there any other questions? No? All right, I'll stop recording. Oh, one question on that. Why is it so eager to get into my agar plates, but it's not taking over the garden?
Do you mean the Aspergillus, Ellis?
Aspergillus.
Yeah. Well, because I mean your agar plates don't have everything else that your soil has to kill them. So your gardens and your soil will have other species of fungi and bacteria that are very adept at killing them. There's a lot of competition.
John's asking.
Yes.
John's asking, what is Cladophilifera only affect males?
So I mean, I think I see Eva in here. She's probably a better person to ask, because I think she's read a lot more about this. I don't know, and I don't actually think anyone does. They also don't really know why it is that it's almost exclusively CNS infections that it causes when it infects people, and no one seems to really know how it is that it's able to get into the brain as effectively as it does. But yeah, it's not sure, but it's a very strange one. It's young, healthy males is the one that it seems to like the most. Or maybe they're just out in the garden more. I don't know, because it's soil borne, and it's so that's sort of where I imagine. But yeah, maybe sport. I don't know.
Very good. Any other questions? All right. So I'll stop recording there.